The world of medicine is on the cusp of a revolution, one that is unfolding faster than ever before. This revolution is driven by a powerful technology called CRISPR-Cas9, which has seen remarkable advances in just the past few years. CRISPR-Cas9 is a gene editing tool that has already moved from the lab to clinical trials, with the first CRISPR-based therapies for sickle cell disease and beta thalassemia approved in 2023 in the UK and US. It allows scientists to make precise changes to DNA, the blueprint of life, and now patients are seeing real-world benefits. This breakthrough has the potential to cure genetic diseases, fight cancer, and has recently been used to develop experimental treatments for conditions like hereditary blindness and muscular dystrophy. Researchers are even exploring CRISPR to combat HIV and to engineer immune cells that can better target tumors, hinting at a future where gene editing could rewrite the story of human health. The story of CRISPR begins in the most unexpected of places: bacteria. Scientists studying bacterial immune systems discovered a peculiar pattern in the DNA of these microorganisms, a finding that won Emmanuelle Charpentier and Jennifer Doudna the Nobel Prize in Chemistry in 2020. They found repeating sequences of genetic code which they named clustered regularly interspaced short palindromic repeats, or CRISPR. These CRISPR sequences were interspersed with segments of viral DNA, a clue to their function as a genetic memory bank. This discovery hinted at a sophisticated defense mechanism, which has since inspired new research into programmable antibiotics and antiviral therapies. Further research revealed that CRISPR worked in tandem with a special enzyme called Cas9, and in 2024, new CAS variants have been engineered to improve accuracy and reduce off-target effects. Cas9 acts like a pair of molecular scissors, and recent studies have shown it can be programmed to target multiple genes at once, opening doors to complex genetic therapies. It can cut DNA at specific locations guided by CRISPR sequences, and new delivery methods like nanoparticles and viral vectors are making gene editing safer and more efficient. When a virus invades a bacterium, the CRISPR-Cas9 system captures a piece of the viral DNA and integrates it into the bacterium's own genome, a process now being mimicked to create living biosensors and programmable cells. This stored viral DNA serves as a memory of past infections and has inspired new research into digital data storage using DNA. If the virus attacks again, the bacterium can use this stored information to recognize and destroy the invader, a principle now being adapted for rapid diagnostic tools. This elegant defense mechanism, honed by billions of years of evolution, has become the foundation for a revolutionary gene editing technology that is now being used in agriculture, medicine, and even environmental conservation. CRISPR-Cas9 gives scientists an unprecedented ability to manipulate the building blocks of life, and with each new breakthrough, the possibilities continue to expand. The beauty of CRISPR-Cas9 lies in its simplicity and versatility. Imagine a word processor that allows you to precisely edit the letters in a document. CRISPR-Cas9 acts in a similar way, but instead of words, it edits the letters of DNA. The process begins with a guide RNA, a short sequence of RNA that matches the DNA sequence scientists want to target. The guide RNA acts like a homing beacon, leading the Cas9 enzyme to the precise location in the genome. Once the Cas9 enzyme reaches its target, guided by the guide RNA, it makes a precise cut in the DNA double helix. This cut activates the cell's natural DNA repair mechanisms. There are two main pathways for DNA repair: non-homologous end joining, or NHEJ, and homology-directed repair, or HDR. NHEJ is a quick and dirty method that often introduces small insertions or deletions at the cut site. This can be useful for disrupting gene function. HDR, on the other hand, is a more precise repair pathway that uses a template DNA sequence to guide the repair process. By providing a template with the desired sequence, scientists can introduce specific changes into the genome. The potential of CRISPR-Cas9 for medicine is vast. It holds the promise of treating a wide range of diseases that were once considered incurable. Already, CRISPR is making significant strides in treating genetic disorders. Sickle cell disease, a debilitating blood disorder, is caused by a single mutation in the gene that codes for hemoglobin. CRISPR-Cas9 has been used to correct this mutation in cells taken from patients, offering hope for a cure. Clinical trials using CRISPR to treat sickle cell disease and another blood disorder, beta thalassemia, have shown promising results. CRISPR is also being explored as a tool to fight cancer. Scientists are developing CRISPR-based therapies that can enhance the body's own immune system to target and destroy cancer cells. CAR T-cell therapy, a type of immunotherapy that involves engineering a patient's T-cells to attack cancer, is being enhanced with CRISPR to improve its efficacy and safety. Infectious diseases are another frontier in the fight against human suffering. CRISPR is being investigated as a potential weapon against viral infections like HIV and hepatitis B. Researchers are exploring ways to use CRISPR to disable viral genes, preventing them from replicating and spreading. The potential applications of CRISPR extend far beyond treating existing diseases. It has the power to transform medicine in ways we are only beginning to imagine. Neurodegenerative diseases like Alzheimer's and Parkinson's, which have long eluded effective treatments, could potentially be cured or prevented with CRISPR. Scientists are exploring ways to use CRISPR to correct the genetic defects that can. So, while CRISPR-Cas9 is an incredibly powerful tool, it does come with its own set of challenges and controversies, you know? One of the main concerns we face is the potential for off-target effects, where CRISPR accidentally edits the wrong part of the genome. Off-target effects happen when the Cas9 enzyme, which is designed to cut DNA at a specific location, accidentally cuts at unintended spots in the genome. These mistakes can have unpredictable and sometimes dangerous consequences. And, well, these unintended edits can lead to unpredictable consequences. That's why there's also a lot of debate about the ethics of using CRISPR, especially when it comes to editing human embryos. For example, the world was shocked in 2018 when a scientist in China announced the birth of the first gene-edited babies. This incident raised huge concerns about safety, consent, and the potential for misuse. It's definitely something we take very seriously. There are ongoing debates about how CRISPR should be regulated and who gets to decide. Scientists are, of course, working diligently to address these issues, both technically and ethically. Improving the specificity of CRISPR-Cas9 and developing clear ethical guidelines are top priorities. One promising approach is engineering Cas9 enzymes with higher fidelity, and another is working with regulators to ensure responsible use. Basically, this means making the technology safer and making sure society's concerns are heard. Another strategy involves developing advanced guide RNAs and ensuring every experiment goes through strict ethical review. These efforts are designed to reduce errors and make sure CRISPR is used safely and responsibly worldwide. It's exciting to see the progress being made, but, there's still work to be done, both in the lab and in society. The goal is to make CRISPR as safe, ethical, and reliable as possible for everyone, everywhere. The power of CRISPR-Cas9 to alter the very fabric of life raises profound ethical questions that have sparked heated debates around the world. The ability to edit the human germline, the DNA passed down from generation to generation, is especially controversial and has led to international outcry in some cases. Germline editing raises the specter of so-called designer babies, children whose genetic makeup has been altered for non-medical reasons, like enhancing intelligence or athletic ability. These possibilities have ignited fierce ethical controversies. In 2018, a Chinese scientist shocked the world by announcing the birth of the first gene-edited babies, sparking outrage and condemnation from the global scientific community. This incident intensified international debates about the risks, responsibilities, and boundaries of CRISPR technology. Meanwhile, the rise of biohacking, individuals experimenting with CRISPR outside traditional labs, has raised new concerns about safety, oversight, and the potential for misuse. The ethical implications of such interventions are complex and far-reaching, touching on issues of consent, equity, and unforeseen consequences. Many scientists and ethicists believe that germline editing should be approached with extreme caution, if at all, and that international cooperation is essential. They argue that it is essential to establish clear ethical guidelines and regulations before embarking on such a consequential path, one that could shape the future of humanity itself. Another challenge facing the field of CRISPR-based medicine is ensuring equitable access to these potentially life-saving therapies. The high cost of developing and delivering CRISPR therapies could make them inaccessible to many patients, especially those in low- and middle-income countries. It is crucial to develop strategies to make CRISPR therapies more affordable and accessible to all who need them. This will require collaboration between scientists, policymakers, and healthcare providers. The field of CRISPR-Cas9 is rapidly evolving, with new discoveries and advancements emerging at a breakneck pace. Scientists are constantly working to improve the precision, efficiency, and safety of CRISPR technology. One promising area of research is the development of base editing, a more precise form of gene editing that can correct single-base errors in DNA without cutting the double helix. Base editing has the potential to treat a wider range of genetic diseases with even greater precision. Another area of active research is the development of new delivery systems for CRISPR components. Viral vectors, which are commonly used to deliver CRISPR components into cells, can sometimes trigger immune responses or integrate into the host genome, raising safety concerns. Scientists are exploring non-viral delivery methods such as nanoparticles and lipid-based carriers, which could offer safer and more efficient alternatives. The next 5 years promise extraordinary progress in CRISPR-based medicine, but also bring new controversies and challenges to the forefront. We'll likely see a surge in CRISPR therapies entering clinical trials and seeking regulatory approval, raising questions about oversight and safety. As CRISPR technology advances, debates will intensify over who gets access, how treatments are priced, and the potential for unintended consequences. We can anticipate the development of even more precise and sophisticated gene editing tools, but also greater scrutiny from regulators and the public. These advancements could pave the way for treating a wider range of diseases, yet they also spark concerns about equity and long-term effects on society. Complex conditions involving multiple genes may become treatable, but the societal impact of such breakthroughs remains uncertain. The ethical and societal implications of CRISPR technology will continue to be debated and refined, especially as regulatory frameworks struggle to keep pace with innovation. We must grapple with the profound implications of this powerful tool, including the risk of misuse and the challenge of balancing progress with responsibility. It is essential to engage in thoughtful dialogue about the societal impact and regulatory challenges that lie ahead, and to establish clear, adaptive ethical guidelines that can evolve alongside the technology. Only by balancing innovation with ethical responsibility can we ensure CRISPR is used wisely and for the benefit of all humankind. Thank you for watching. If you enjoyed this video and want to stay updated on the latest in science and technology, please like and subscribe. Your support helps us bring you more fascinating content.